Welcome to the Deep Dive. If you're here, you're probably looking for a shortcut to expert knowledge. And today we are diving into a question so fundamental that, well, scientists have been arguing about it for centuries. How do you define life? It sounds so simple. I mean, we know it when we see it, right? But pinning down the actual necessary and sufficient conditions for something to be classified as alive, that might just be the single hardest challenge in modern science.
It absolutely is. And what's really fascinating here is that this isn't just a question for biologists anymore. Our source material for this really hammers that home. It draws a lot from the astronomer Joseph Shklovsky.
An astronomer. That's an interesting perspective.
Exactly. He was an astrophysicist, and he was trying to figure out the probability of life elsewhere in the universe. But he realized he couldn't even start that calculation because he didn't have a solid definition for life here on Earth. He admitted right up front that the origin of life is still, a huge unsolved mystery. We just don't know the exact conditions on early Earth.
Right. And if we get bogged down trying to figure out how life started 4 billion years ago, we'd be here forever. So our mission today is to sort of sidestep that a bit. We're going to focus on what life is, not necessarily how it began. And we're going to look at it through two very different but ultimately connected frameworks. First, the classic material view, the biochemistry, the molecules. And second, a really revolutionary, more abstract view based on cybernetics and control systems.
Okay, so the starting point, the classic framework, was heavily influenced by the academician A.I. Oparin back in the mid-20th century. Oparin really zeroed in on the material complexity. He said life requires these complex molecular clumps, mainly proteins, that show what he called ordered metabolism.
And ordered metabolism. Just to be clear for everyone, that's basically the continuous structured exchange of energy and substances with the environment. So you're eating, breathing, excreting, that's metabolism.
That's the intuitive idea, yeah. But if you lean on just metabolism, the definition starts to get leaky really fast. And the source material points this out immediately.
Yeah.
Metabolism is a fundamental attribute of life, for sure. But it might not be the essence. It's not the one thing that separates life from non-life.
Okay, so why not? Where does that simple definition start to fall apart?
Well, the source notes that you can actually see basic forms of substance exchange in things that are definitely not alive, like certain oscillating chemical reactions or simple colloidal solutions. They can take up and release substances in a semi-ordered way. Oparin's early work on coacervate droplets, these little blobs of organic molecules, showed complexity, but they were missing the key ingredient.
Which was?
A system of control. And just as important, a system of replication. So to really find the essence, we have to go deeper than just the exchange of matter. We have to look at the specific structures that govern that exchange, the material carriers, as the source calls them.
And that's our way into the material view. We're moving from general chemistry to the really precise architecture of biology.
Yeah.
So let's talk about the two big ones, the fundamental material carriers of life, proteins and nucleic acids.
Let's start with proteins. I mean, these things are just engineering marvels. They're huge, incredibly complex macromolecules. Their atomic mass can get up to 10 million units. They make up most of the machinery in a cell, the enzymes, the structural bits, pretty much everything that does the work. And they're built from these simpler little subunits called amino acids.
And this is where we hit our first absolutely astonishing detail. It really highlights how unique life's chemistry is. Organic chemistry knows about, 100 different amino acids that occur naturally?
Roughly, yeah.
But pretty much all life on Earth, from a tiny bacterium to us, uses only 20 of them to build all its proteins.
It's an incredible specificity. And you have to ask, why just those 20? And one of the leading ideas is just efficiency and stability. These 20 amino acids are like a perfectly optimized chemical toolkit. They have all the properties you need. Some are water-loving, some are water-fearing, some are acidic, some are basic. And that allows them to fold into these incredibly complex, stable 3D shapes. And there's another layer to it. Terrestrial life uses what we call left-handed versions of these amino acids. That consistency is critical.
Why is that so important?
Because if you start mixing left and right-handed molecules, the whole protein structure becomes unstable. It just doesn't work. So life basically chose a small, specific, very efficient toolbox and then committed to it completely.
Okay, so proteins are the structure, the workers. They do the stuff of life. But proteins are also pretty delicate. They break down all the time. They're the machinery, but they're not the permanent memory. So if proteins are the factory, what's providing the perpetual blueprint and the automated builders you need for this constant cycle of destruction and reproduction?
And that brings us to the famous pair, the nucleic acids. Indeed, DNA and RNA deoxyribonucleic acid and ribonucleic acid. These are basically long polymer chains made of repeating units called nucleotides, and they are the information management system of the cell. They hold all the instructions for building those proteins, and they are the absolute basis of heredity and reproduction.
And their structure is everything. We have to talk about that revolutionary 1953 discovery by Crick and Watson. It basically launched the entire field of molecular biology.
It was a huge moment of synthesis. They were using X-ray data, mostly generated by Rosalind Franklin, actually. And what Crick and Watson figured out was that DNA isn't just a single strand. It's two strands, paired up and wound around a central axis. That's the iconic double helix. And to give you a sense of scale here, the diameter of this helix is ridiculously small. We're talking 18 to 20 angstroms.
And an angstrom, just for context, is 1/10,000,000th of a meter. So we're talking about atomic dimensions here. The pitch of the helix, one full turn, is only 34 angstroms. It's an unbelievably compact way to store information.
And the real breakthrough, the true genius of it, wasn't just the spiral shape. It was understanding how those two strands are connected. This is the pairing rule.
Let's talk about that rule because it's the foundation of everything we're about to discuss.
So the two strands are held together by these weak little forces, hydrogen bonds, that connect the bases jutting out from the backbone. And the bonding is incredibly specific. Adenine, or A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, Ca with T, G with C, always.
And that specific pairing does two things, right?
Exactly, two things. First, it makes the molecule stable. And second, it means that when you unzip the strands, each side contains the perfect template to rebuild its missing partner. It's the mechanism of perfect replication, right there at the molecular level.
Okay, so this brings us from structure to function. We have this beautiful spiral molecule. But what does that specific paired structure actually do? Why is it considered the material essence of life?
Because its main job is the highly stable, highly reliable hereditary transmission of genetic information. The DNA molecule is the master program. It's a code that controls the exact sequence of amino acids for building every single protein in the cell, which means all the inherited traits you can think of, your eye color, the specific shape of your liver enzymes, it's all determined by the sequence of those ATGC pairs along the DNA.
So the secret is in the sequence, and the person who first realized this was G.A. Gamow, who, as you said, was this brilliant mind who worked on everything from nuclear physics to the Big Bang. He looked at the structure in 1954 and realized it had to be a cipher. It had to be a code.
And Gaimow's contribution here was purely theoretical, but it was just so elegant. He didn't know the code, but he understood the constraints of the problem. He just used pure logic. The puzzle was simple. How do you code for 20 different amino acids?
The protein building blocks.
Using a system that only has 4 unique letters. The bases A, G, T, and C.
Okay, let's do the math here slowly because this is so cool.
Right. So if you try one base for one amino acid, you only have 4 options, A, G, T, C. Not enough for 20. So what if you try combinations of 2 bases?
Okay, so like AA, AT, AG, AC, that would be 4 times 4, so 16 combinations.
16. Still not enough. You're 4 short.
So the minimum unit of information had to be a group of 3 bases, a triplet, which we now call a codon.
Precisely. If you use a triplet, a combination of 3 bases, you get 4 times 4 times 4, or 4 cubed. That's 64 possible combinations, which is more than enough to code for the 20 required amino acids. And that redundancy is actually a key feature. It means multiple different triplets can code for the same amino acid, which gives the system a little buffer against errors.
And Gamow's brilliant bit of logic was confirmed experimentally just a few years later in 1961 by Nierenberg and Mathai. They basically built proteins in a test tube.
They did. Their experiments were amazing. They created a synthetic piece of messenger RNA that was just a string of uracil bases, UOU. They put it in a test tube with all the necessary cellular machinery, and they saw what protein it made. And it produced a protein made entirely of one amino acid, phenylalanine.
That was the first word of the code ever deciphered. UU codes for phenylalanine.
It was. And the absolutely astonishing conclusion that came from all the work that followed was that this genetic code, it was basically identical across all known life on Earth.
So it's the unifying language. From blue-green algae all the way up to us, we all use the same biological dictionary.
Yeah.
Although you mentioned a small footnote in our source, right? It's not absolutely 100% universal.
Right, and that's an important point for accuracy. Later research did find some very minor variations, mostly in mitochondrial DNA or in a few species of single-celled organisms. But the fundamental structure, the language of the triplet codon, it's standardized. And that standardization strongly implies a single common ancestor for all life on Earth, even if a few dialects have popped up since then.
Okay, so now we get the blueprint, which is the DNA, and we get the language, the triplet code. But how does a cell actually turn that code into a physical thing, into a protein? We need to talk about the logistics here, the protein factory. And that means we have to talk about RNA.
Right. RNA, or ribonucleic acid, is the essential go-between. It's a smaller molecule, molecular mass of around 10 to the 6th, so much smaller than the huge DNA chains, and it's different from DNA in two key ways. It uses a slightly different sugar in its backbone, and it swaps out one of the bases. It uses uracil U instead of thymine T, and the source describes this whole process of protein synthesis as this incredibly precise automated factory that makes our most advanced manufacturing plants look clumsy.
Okay, so walk us through the assembly line, from retrieving the information to the final product.
It all starts in the nucleus with a process called transcription. The information in one specific section of the DNA, a gene, gets copied onto a complementary strand of informational RNA, or mRNA. Think of the mRNA as a disposable working copy of the master blueprint. Then, that mRNA leaves the safety of the nucleus and travels out to the sites of protein assembly, the ribosomes. And the ribosomes themselves are these complex machines made mostly of another type of RNA, ribosomal RNA or rRNA.
So the mRNA is the instruction sheet at the factory. Who brings the raw materials, the 20 different amino acids?
That's the job of transport RNA or tRNA. And you have to have at least one unique type of tRNA for each of the 20 amino acids. The tRNA molecule is a really clever little adapter. On one end, it carries a specific amino acid, and on the other end, it has a 3-letter sequence called an anticodon. So when the tRNA gets to the ribosome, it matches its anticodon to the correct 3-letter codon on the mRNA strand, like a key fitting into a lock. And this programmed sequential assembly makes sure that the amino acids are linked together one by one in the exact order specified by the original DNA code. And you get a perfectly formed functional protein. The speed is just phenomenal. In bacteria, they can add up to 20 amino acids per second.
That's an incredible level of programming. But the source throws in a really fascinating wrinkle here. It talks about the discovery that the genetic text isn't just one long, continuous sentence. It mentions a mosaic structure. What's that about?
This is a future mostly of more complex life, eukaryotes. Researchers discovered that the DNA text is, well, it's like a mosaic. The meaningful coding pieces, we call those exons, are often separated by these long, apparently senseless fragments. We call those introns. These introns don't actually carry any information for the final protein.
Why are they there? What are they doing?
That's still a hot area of debate, actually. But the existence of this genomic mosaicism means the process isn't just a simple copy. When the mRNA is first copied from the DNA, it includes a critical editing step called splicing. These unnecessary non-coding intron pieces have to be precisely cut out. And then the remaining coding exon pieces have to be stitched back together perfectly before that final, mature mRNA can go to the ribosome.
So it's not just photocopy the blueprint. It's photocopy, get out the scissors, cut out all the junk, and then tape the useful parts back together. It makes the whole thing seem so much more complicated.
It does. And the source notes that the origin and function are still basically unknown, but it adds this huge layer of control and potential regulation. Some people think introns help speed up evolution by letting you shuffle the useful bits, the exons, into new combinations. Others think they might just be ancient junk DNA that the cell has learned to work around.
Okay, so we've established this incredibly sophisticated DNA system. It's basically a high-fidelity cybernetic device designed for one thing: identical reproduction, for stability.
Right.
But if life is all about stability, how does evolution, the constant change of life, actually happen?
That's the paradox. The system is built for stability, but stability in life has to be balanced with a capacity for change. Otherwise, you just hit an evolutionary dead end. And the DNA system, even though it's protected by all these repair enzymes and proofreading mechanisms, it's not completely foolproof.
And those occasional mistakes, that's what we call a mutation.
Exactly. External factors, things like hard radiation, certain chemicals, or even just random errors during the copying process itself can cause these little disturbances or errors in the hereditary code. And this is the crucial point. These errors lead to brand new heritable traits in the offspring that weren't there in the parents.
So the system is fundamentally stable, which lets the species continue, but it's also statistically prone to these tiny random errors. And those errors are the raw fuel for evolution.
That's it, exactly. And we have to stress these mutations are random. They're almost always useless or even harmful. And that's where Darwinian natural selection comes in. Nature applies what the source calls a very rigid selection. Organisms with bad mutations get weeded out, but an organism whose random mutation results in some beneficial new trait, maybe a slightly better enzyme or resistance to a disease, that organism survives, it reproduces, and it passes that new, slightly altered blueprint on. That's the engine of evolution.
And the really crucial takeaway here, which our source highlights, is that those older theories about the origin of life, the ones focused on just the chemistry of the primordial soup, they were missing this piece. They could explain the ingredients, but they couldn't explain the instruction manual.
They understood the basic chemistry, yes, but they completely missed the programming. They missed the mechanism for heredity, for variability, and for the sustained self-reproducing complexity that depends on that information system.
So this molecular definition life is carbon-based. It's built on proteins and DNA. It uses a triplet code. It's incredibly powerful for describing life on Earth. But now we get to the big pivot point. Why isn't that definition enough, especially when we start looking beyond our own planet?
Because it's too specific. It's too Earth-centric. If we're talking about the possibility of life on other planets, which is the whole motivation for the work we're looking at, we can't assume it will follow our chemical rules. What if alien life uses silicon instead of carbon? Or a different solvent? Or a totally different information polymer?
We need a definition that strips away the specific chemistry and gets to the core of what life does, the functional requirement. And this is where the famous mathematician and cybernetics expert, academician A.N. Kolmogorov, comes into the picture.
Kolmogorov was adamant about this. He pointed out that biology has really only ever studied one thing: organisms on Earth that all share a common ancestor. To find truly different forms of life out in space, maybe something plasma-based or something based on physics we don't even understand yet, we need a universal framework. We need an approach that focuses on what life does. Its organizational properties, its control properties, not what it's made of.
A purely functional definition. We're shifting from molecules to systems theory. And this work was really spearheaded by the research of A.A. Lyapunov, who applied the new field of cybernetics to biology.
Yeah, cybernetics, the science of control, communication, feedback loops. It was developed by Norbert Wiener in the 40s. And Lyapunov realized that you could translate all these biological concepts into a universal language of control.
So, for example, instead of talking about DNA, you talk about information storage.
Okay.
And instead of irritability, like an organism reacting to something, you talk about feedback loops and coded signals.
Exactly. Lyapunov's starting point was this: control is the most fundamental characteristic property of life, no matter what it's made of. Life, at its core, is a control process.
And the state that life is trying to control, to maintain, is what Lyapunov called relative stability.
This is the central concept. So any substance has its physical characteristics, mass, energy, composition, and so on, and these things naturally change. Lyapunov says a substance is relatively stable if the average values of those characteristics change very little over a long time, especially when you compare it to similar non-living things in the same environment.
Okay, so stability in the face of change. And that stability can come from two places. It can be passive or it can be active.
Right. Passive stability is easy. A diamond is stable because its chemical bonds are super strong. It just sits there. It takes a huge amount of energy to change it. But active stability, that's what defines life. Life's stability comes from a constant fight. It comes from internal reactions that are actively trying to preserve its state against a changing environment. Lyapunov called these preserving reactions.
And these preserving reactions are the basis of life's incredible adaptability. It's not just surviving, it's actively maintaining a very specific, complex internal state.
Yes, absolutely. So when your body temperature goes up, you initiate a preserving reaction, you sweat to cool yourself down, you're maintaining that specific internal equilibrium that your enzymes need. And the source points out that it's not just passive adaptation either. Organisms actively transform their environment. I mean, think about the first photosynthetic organisms that pumped our early atmosphere full of oxygen. That was a massive planet-scale preserving reaction that made all aerobic life possible.
And this huge library of preserving reactions is all managed by a very sophisticated, information-based cybernetic control loop. Let's break down the mechanics of that loop.
Okay, so it's a continuous multi-stage process. First, input. The system has to get information about the outside world through coded signals, and Lyapunov stressed that these signals have to be discrete.
Meaning they're digital, not analog, like on or off.
Exactly. They have a finite number of values, which makes them reliable to process. In biology, that could be a neuron firing or a hormone binding to a receptor. Second, processing. This information gets analyzed in a managing system. In simple life, it might be a biochemical feedback loop. In complex life, it's the central nervous system. Third, comparison and storage. The managing system compares this new information to data that's already stored in a memory device.
The DNA, the inherited information.
The DNA. And the more flexible the system needs to be, the more environments it can handle, the larger its memory has to be. And fourth, output. Based on that comparison, the system sends out new signals, and those signals trigger the internal changes, the preserving reactions that fight against entropy and maintain that stable state.
That model is so abstract, it could describe a computer just as well as it describes a cell. But how does that cybernetic need for a reliable memory get us back to the physical world? This is where Lyapunov makes this incredible connection between systems theory and quantum physics.
It's the requirement for something to be both very small and extremely reliable. You need a huge memory for flexibility, but that memory has to survive the constant jiggling of heat.
Small but stable. How do you get that in the material world?
Lyapunov concluded that the only things that work are individual molecules made of a large number of atoms. The stability comes from quantum mechanics. To change the state of a big molecule, to flip a bit of information, you have to put in a significant amount of energy, specifically more than 0.1 electron volts.
And that number, 0.1 eV, that's the magic threshold, isn't it?
It is. The energy needed to break or seriously alter a strong molecular bond, like a hydrogen bond, is right in that range. And here's why that's critical. The energy of random thermal motion, the constant bumping and vibrating of atoms from heat, is significantly less than 0.1 eV.
Ah, so the random thermal noise that's everywhere that would corrupt a weaker storage system, it just isn't strong enough to accidentally flip the bits of information stored in these big, complex molecules.
Exactly. The memory molecule is effectively insulated from thermal chaos by its quantum nature. And this physical requirement for stable quantum-based information carriers is the final piece of the puzzle. It doesn't say it has to be carbon, but it naturally leads you to high molecular weight polymers like proteins and nucleic acids because they are exactly the kind of structures that meet that stability criteria.
Which lets us finally state Lyapunov's complete functional definition, which is totally independent of any specific chemistry. Life is a highly stable state of matter that uses information coded by the states of individual molecules to generate preserving reactions.
It's an engineering definition, and it finally explains why the double helix is so effective without saying that life has to use a double helix.
And this definition immediately brings us face to face with the laws of physics, specifically thermodynamics. If you're a highly stable system that's actively fighting the universe, what are your energy requirements?
You're fighting the second law of thermodynamics, the law that says disorder or entropy always increases in a closed system. So to maintain your complex, organized, stable state, you have to constantly work against that natural tendency towards chaos.
Which means you need a continuous input of energy to counteract those thermodynamic processes that are trying to break you down.
Precisely. If a living thing were a closed system, its entropy would just go up and up until it died. So a living organism has to be an open system. It has to constantly take in energy from the outside, and just as critically, it has to systematically remove the entropy that accumulates inside it.
And this brings us right back to the beginning. What is the observable process that accomplishes this energy intake and entropy removal?
That process is metabolism. So under this cybernetic view, metabolism isn't the definition of life. It's the method that the control systems use to regulate the stable state and manage entropy. You have to eat low-entropy stuff, like food, and you have to get rid of high-entropy stuff, like heat and waste products.
That makes the problem with those older operant-style views so clear. They were mistaking the symptom metabolism for the root cause, which is the need for stable information control to fight entropy. Metabolism is just a necessary consequence of being a highly organized control system in a thermodynamic universe.
And that control system isn't just one big thing. A key feature of life is that it's organized into these discrete units: cells, organs, organisms, populations, species. And that structure reflects a hierarchical system of control.
So information is being managed at multiple levels at the same time?
Yes.
Yeah.
The higher level systems are constantly adjusting and tuning the state of the lower level systems. Think about your own body. Your brain, a high-level system, regulates the temperature of your liver, a lower-level system. And Lyapunov breaks this down into two main types of control: structural and statistical.
Okay, let's spend a minute on that distinction. It seems important for understanding stability over different timescales. What's structural control?
Structural control is what happens inside an individual organism. It's fast. It's a multi-layered management system with a clear chain of command. Your nervous system, your endocrine system, perfect examples. These systems act very quickly. Homeostasis, healing an injury, a fight or flight response. That's all structural control. Each part acts on the part below it while submitting to the slower, higher-level parts above it.
And statistical control.
That governs the things above the individual organism: populations, species, ecosystems. It involves a huge number of independent, statistically equal units, the individual organisms, all interacting randomly through competition, cooperation, and so on. And this type of management is inherently slow-acting compared to structural control. It deals with long-term evolutionary trends.
That's a brilliant distinction. So an individual organism reacts instantly to stay in balance. That's structural control. But a whole species adapts to climate change over thousands of generations. That's statistical control.
And this hierarchy leads directly to an interesting outcome. Superorganismic things, like a species, are way more stable and live much longer than the individual organisms that make them up. The individuals are designed to perish quickly. The system preserves the information, the type, not the specific machine.
Which brings us full circle to why reproduction is so necessary. If individuals are always dying off, the species has to constantly replenish its members. And the key ingredient for making a new, stable organism isn't just matter, it's information.
Exactly. It is statistically impossible for that huge, complex library of information, the entire control system you need for all those preserving reactions, to just spontaneously assemble in every new organism.
So the new organism has to get its entire information stock, the code, the blueprints, the operating system, in a ready-made package from its parents.
And this is the great moment of synthesis. Reproduction is the self-reproduction and transmission of information. This is where Lyapunov's abstract cybernetics perfectly merges with the concrete reality of molecular genetics from Crick, Watson, and Gamow.
The incredible stable diversity that you can create with the 4 DNA bases and the 64 codons. That is the stock of information being transmitted. The DNA molecule is the stable information carrier that was selected by those physical criteria we talked about earlier.
And if we're going to think about life as an information transfer system, we have to talk about errors. That's a standard part of any communication channel. Information transfer always happens against a background of interference, of noise, that can distort the message.
And in heredity, the noise that distorts the message.
Yeah.
That's mutation.
Correct. A mutation is just a distortion of the inherited information during the transmission process. So from the cybernetic viewpoint, a mutation literally changes the control system itself. And that change alters the organism's set of preserving reactions, which in turn changes how it interacts with its environment.
And that variation in how it interacts with the environment radically changes its probability of survival. This unified view takes something that was a grand observational theory, Darwinism, and it lays the foundation for a rigorous mathematical theory.
It lets you analyze the mechanisms of evolution without caring if the life form uses DNA or RNA or some weird alien polymer. If you have a system that can self-reproduce coded information, it is going to be subject to hereditary variability, which is noise or mutation and natural selection, the survival test imposed by the environment. Lyapunov's concepts are really seen as the first draft of a truly rigorous mathematical theory of Darwinian evolution that finally incorporates molecular mechanics into the equation of control.
So today we have traveled from the very specific chemistry of 20 amino acids and a 34 angstrom pitch, all the way to this profound abstract definition of life as a molecular control system fighting entropy. And the source material makes it really clear that the final complete understanding of life isn't here yet. But when it comes, it's going to require a permanent fusion of these two great ways of thinking.
That ultimate understanding will come from the synthesis of advanced cybernetic concepts and deep biophysicochemical knowledge. This comprehensive approach, bringing together the functional principles of Lyapunov and Kolmogorov with the material reality of molecular biology, is absolutely vital. Not just for figuring out how life started here, but for guiding our search for life across the entire universe. Because if our definition is too narrow, we are guaranteed to miss the truly alien life forms that might be out there.
And here's something profound for you to think about as you go about your day. If life is fundamentally defined as a highly stable state of matter, using coded molecular information for self-preservation against the forces of entropy, what radical non-carbon-based materials could also meet that functional definition? Could a self-organizing system of energy flow, maybe something involving superconducting circuits or magnetic domains, a system with no DNA at all, still have the stability, the memory, and the preserving reactions to be considered alive under this demanding cybernetic model? That's the question that will define the future of astrobiology.